All right. Good afternoon. My name is Andras, and I'm from a small yet very proud country in Central Europe called Hungary. And just to be clear, no, I'm not hungry. And yes, I only eat goulash and paprika. So now that we got that out of the way, I work as an AI engineer for Phenotyping, and my background is mainly in computer science, graph theory, discrete mathematics. So, the stuff that typically has nothing to do with plants. And at Phenotyping, I was lucky to participate in several projects where I learned a little bit more about plants and more specifically, more about the processes that are typically involved in industrial plant phenotyping. By talking to our customers and visiting our clients, we see and understand that space is a very precious, expensive, and limited resource. And because of that, in these facilities, choosing for the integration of some sort of a sensor-to-plant imaging system is often an attractive solution. However, most of the sensor-to-plant imaging systems will have some sort of a limitation about the density of the plants that they can deal with while still extracting individual plant parameters. And because of that can be a problem because that has a direct impact on the capacity of the facility and it also has a direct impact on the throughput. And we at Phenotyping really believe that these are exactly the kinds of problems we need to solve as an industrial supplier to be successful in driving our mission forward to make digital phenotyping the standard. And to address this problem, we have developed a method called GridCast, which ingests a 3D point cloud, just like this one in the image. And in a first step, the algorithm performs a 3D plant instance segmentation on this point cloud. That means that the method annotates the vertices of the point cloud to convey information about which points belong to which plants exactly. And I guess this is still quite easy to understand why this is absolutely necessary, because if we wouldn't be able to tell the plants apart in the data, we wouldn't be able to extract plant parameters on the individual level in the first place. Then in a second step, the algorithm extracts an underlying grid-like structure that best represents the arrangement of the plants. And this grid-like structure that we often refer to as the plant grid or the phenotyping grid is a very typical arrangement of plants in phenotyping experiments. And I guess this is not very easy right now to appreciate why this problem is difficult and why it's important to solve it, because you could say, well, as a human, when I look at this image, I could very easily count how many rows and columns are there, and I could possibly even just draw in this underlying plant grid. However, if I take the plants away, that's a bit slow, maybe it's a little bit easier now to see why this problem is not so straightforward to solve. And then in a third and final step, then the algorithm also assigns the detected plant instances to positions on the grid, which you can see denoted here by the column and row numbers in the image. So before I delve into the technical details behind the approach, I would like to note that we have taken a slightly different route to what we nowadays see in academic research, where the trend exhibits an increased focus on the development and application of more and more sophisticated, more and more complex machine learning architectures. And these are really interesting. However, when we would like to develop something for the industrial scale, and ultimately we would like to turn our solution into a product at some point that's robust and reliable, there are a couple of things to keep in mind. First of all, ideally, the method that we develop is easily deployable on common hardware. Typical scenario that we would face is that we often need to install our entire software stack on an old Linux server that's been sitting in the greenhouse, and this directly disqualifies all the resource-intensive and typically GPU or edge hardware-dependent approaches for us. And moreover, because in phenotyping, both the plants and the experiments are just so very, very diverse, it's very difficult to just develop a single model to rule them all. And because of that, we really wanted to develop something that is adaptable and can be quickly and easily trained on a per experiment basis. So I can tell you from experience that if you train a model that has a good performance on, let's say, corn, in a very short while, you can be absolutely sure that there will be people asking you if we can use the same thing now on Christmas trees. And typically the answer is no. Unfortunately, we need some kind of an adaptation. This is what we sometimes also refer to as the problem of domain shift. And I would like to show you the approaches that the pipeline that we developed in technical detail, and take you through the journey that the data takes through the system. And it all starts here. This is a typical setup at Phenospec. This is a multi-view, multi-spectral, high-resolution 3D scanner system. This one is called the Field Scan, and it's equipped with a pair of Plantai F600 3D scanners. And the data that comes out of this setup is a point cloud that already contains fused spatial and spectral information. And I can say as a developer, this is kind of a luxury to already be able to start with a rather powerful data representation. And instead of taking these point clouds and now directly gushing them into some machine learning model and then hoping for the best, we have come up with a compact 2D data representation approach. So the 3D point cloud first is flattened into a 2D projection image where the depth information is just treated as an additional color channel. And then through statistical analysis, we have realized that actually 3 main axes are enough to explain most of the variance in the data. And we have chosen NDVI, GLI, and depth as the 3 main axes to encode the information. You could ask the very valid question now that, okay, how can we perform complete 3D segmentation based on these 2D projections? Are there not going to be stuff that we are missing? And to understand that, it's important to note that these 2D projections are generated from the view of the scanner, which then results in a direct one-to-one correspondence between the 2D pixels of the projection and the 3D vertices of the point cloud. This is a very desirable situation because it means that any kind of annotation extraction we perform on this 2D projection, which is much cheaper than doing it in 3D, will be directly translatable to the 3D point cloud without the loss of any data or without the loss of any resolution. And this really just allows us to open up our trusted, industry-proven 2D instance segmentation framework. That's exactly what we did. We chose a YOLO variant for this task that generates both bounding boxes and segmentation polygons. But I'm not going to talk about the object detector in more detail because these are very well described in literature. But even though these segmentation frameworks are very well known and are proven, the successful use of these frameworks is still dependent on the availability of a large volume of training data, of course, and the domain shift. So, the whole corn versus Christmas tree thing is still a problem. So to address these problems, we are using methods called weak supervision and self-training. And instead of giving you the theory, I will show you how it works in practice. So as step 1, we generate some initial training data set in which the plants are placed far from each other. And we just do this by using our physical setup. And we'll call these the easy instances. Using these easy instances, then we can extract a pool of plant units using deterministic methods like color filter, simple stuff. And this is what we refer to as weak supervision because we are not doing any exact manual annotation. We are just using some heuristic methods to then build up our plant unit pool. And then we can use this extracted pool of plant units to now generate increasingly more and more difficult training instances. And we do this through just standard data augmentation processes that are well known in image processing. So now as we are generating these more and more difficult examples on the fly, instead of directly feeding them into our network, first we perform a zero-shot detection on a batch that we have generated. And we only add the ones to our training set, which were already predicted with a high confidence and with a good result. And this is what we call self-training, and this is immensely useful in applications when you would like to make the training process as automated as possible. And because your training curves will be a lot more predictable, the stopping conditions will be determined much more easily. And this is, again, a very desirable outcome because now we can train an instance segmentation model that works in 3D relatively cheaply and we don't need to do any kind of manual annotation. And also because of the whole self-training stuff, the training regime, we are effectively abstracting away all the machine learning complexity from the user. And we would really like our users to be focused on the phenotyping and not sitting in front of the screen drawing bounding boxes. So that concludes the part, the description of our solution to the instance segmentation problem. So now I will be addressing the grid detection and the plant-to-grid assignment. So as I said before, in phenotyping experiments, typically the plants are arranged in some sort of a 2D grid. And as we have seen before, this might not be the most straightforward problem to solve. But what is the difference between here, the problem seemingly being difficult, but here we can see the grid pretty easily. So perhaps we could ask the person with no pens, and he could perhaps say that, well, he sees where the 2D grid is because he's looking at the setup and his brain is able to find reference points, and that's how he sees where this plant grid is really located. And but there are no reference points, and this is exactly the thought process that we followed when we realized that detecting anchor objects and reference objects can immensely aid the grid detection. Right now you can see it's that's much easier to detect the grid. So we know where the plant grid is located at this point, but we still need to figure out which detected plant instance belongs to which grid cell exactly. And this is non-trivial. Again, as a human, you could say, well, I start at the corners and I make my way through this grid. But there's a problem with that, because as soon as you make a mistake, that can potentially disrupt the assignment of other plants too, to their correct grid cells. And perhaps I could illustrate this better with an analog that's called the dinner party seating problem. So, dinner parties, typically at traditional weddings, there is a very strict etiquette on who should sit where. But let's say there's this one person who just really doesn't want to sit next to their mother-in-law and requests to be seated somewhere else. And you as the party organizer would like to honor this request, but at the same time, you would still like to stick to the protocol as much as possible. And you will soon realize that you have to replan the entire arrangement around the table to accommodate for this request. And in computer science terms, we would say that we are looking for the globally optimal solution, or that we are solving a global optimization problem. And that is what is common with the plan-to-grid assignment problem. And linear programming has been a very well-researched, very well-established field in computer science and mathematics. And it is very powerful at solving problems which can be formulated with a linear objective function and a linear set of constraints. And that's why it's heavily used in business process optimization, in planning and scheduling, operation research, and so on, where finding this globally optimal solution is very critical. And I understand that if you don't have a background in computer science, this might not be the most approachable thing. But the point here is that we can formulate the plant-to-grid assignment problem as a linear program that can be efficiently solved with existing methods. So if we extract the global solution now from the linear program, we know that we have a solution that is optimal and there is no better way to arrange the plants. So now we could also start to infer where maybe the missing plants would be, and so on. In our latest experiments, we were busy with trying to link the performance of GridCast to, which you can see here, evaluated across species. And as you can see, we're still working on the Christmas tree. But what we are really focused on is trying to link this performance to then the impact on the plant parameters and the impact on, okay, what does this now mean for our spacing exactly? And in this graph, you can see the R-squared on the y-axis, which is the correlation between the extracted digital plant parameters and the ground truth data that was manually measured. And on the x-axis, you can see the spacing ratio that starts at 1, and that means no overlap between the plants. And as we move over to the right, at 0.75, the spacing ratio translates roughly to 25% overlap between the plants. And at 0.5, this means a 50% overlap. And as you can see, our baseline parameter extraction a pipeline that is marked with a blue line here has already a very good correlation with the ground truth data when the plants are not overlapping. But as you can see, as we start putting the plants closer together and we move over to the right, this starts to decrease. And it doesn't fall apart completely, but there is a significant drop. And if you use GridCast, then up to about 0.75 for spacing ratio, we can maintain the same data quality. And maybe this is not very easy to understand at this point. What does this mean exactly for the spacing? But this move from a spacing ratio of 1 over to 0.75 roughly translates to doubling the throughput of a phenotyping area. And imagine now how nice that would be to screen the plants twice as fast or put twice as many plants under your imaging system. So before concluding the talk, I would like to just note that we believe that we can really benefit from an efficient workflow in which we take complex problems, we translate them into forms where well-established methods already exist, and then we solve the problems in those domains. And this effectively enables us to thrive by using leveraged methods, and we can much more easily serve our design goals. I just would like to give a shout out to our application scientists, Martina Lazarin and Max Peters, who are the real people behind doing all the legwork of growing the plants, watering them, scanning them, measuring them, and ultimately then archiving the data for people like me to use. That's why I like to call them the data fairies, because they just appear at some point and the next day I have data to work. So thank you very much. And if we still have some time, then I'll be taking some questions.
You got that? What? That? A lot of elegant work to some simple problems here. One of the ones I was really excited about was you're representing depth as part of an image. Can you say more about that?
So it's really just taking the, what comes out of the 3D scanner is a 3D point cloud, right? And there, if you orient the point cloud in a way that Z is pointing up, then essentially you can turn it into a height field, which just becomes an extra color channel in your 2D image.
So it's just the vertex. It's not the whole plant. It's the single vertex of the apex.
So you have a vertex with XYZ coordinates, right? And then you just turn the Z coordinate into color information. And then the whole thing becomes flat. And yeah, that's pretty much. Does that answer your questions? Yes.
I may miss your explanation, but could you please make an explanation about the spacing ratio?
Yeah, sure thing. So the spacing ratio, well, I said that it translates to roughly 0.25 at 0.75 of a spacing ratio. This roughly translates to 25% overlap between the plants, but this is actually an oversimplification. So spacing ratio is calculated as the measuring the average diameter of the plant and then dividing that by the center-to-center distance of the planting positions. So a spacing ratio of 1 actually means that the plants are exactly as far apart from each other as big we expect them to be. So ideally, they would be just not touching.
From grid design to anchor design. So your last slide. Next one.
Next one. Yes.
Grid design to anchor detection. What does anchor detection mean?
So we did the anchor detection to then be able to get some additional information about the images that makes the grid detection a lot easier for us. And in this particular case, the anchors corresponded to these planting cubes or the pots that were in the images. But in theory, these anchor objects can be anything that is easily detectable and conveys some information about an occupied grid cell. Yeah, 5 minutes. Here.
Thank you for presentation. My question is, you said one challenge is about the high volume of the annotation data set. Yes. Have you using transfer learning method or data augmentation for solve this problem?
So we are using data augmentation in terms of using the extracted plant unit pool to then generate increasingly more difficult instances. And the idea here is really that when the instance is easy, when the plants are far from each other, this problem is pretty easy to solve with very simple methods. And for that, we don't need any machine learning or any kind of sophisticated approach to deal with that. The problem becomes increasingly more difficult as the plants getting more and more intertwined, where we have to resort to something a little bit more sophisticated. And then the good thing about it is that we don't have to annotate either of them. And yes, transfer learning could help in a way. And we are using transfer learning in the backbone of the detector, for example. So there you do use a pre-trained backbone for the network that we then tune towards detecting plants.
You work about ULV8, yeah?
Yes.
Okay. Yes, because I work about ULV8 and transfer learning get a better performance. That is why I asked you, did you work about transfer learning, or because this is a good solution for get a better performance?
Yes. Of course, these pre-trained backbones, these networks are really good at detecting features like corners and shapes, which are then, yes, we don't have to relearn that, so the training process is also faster.
Okay, thank you so much.
One last question.
Thank you. I'm really interesting about the self-learning, and you say you didn't get a well result, you will go more data. So do you face any overfitting if you pull more data and reduce the result? How do you balance how many data you pull, how many data generation?
I'm not sure if I got that.
Yeah, because you mentioned the self-training. If you not get the prediction very well, you will make more data for continue the training.
Yes.
And usually put more data, maybe facing the overfitting problem.
Yes, so with the self-training idea, we have direct control over the increasing difficulty of the instances. So, right, if we would just try to generate the very, very difficult ones immediately, then you would also see these spikes in the training in the learning curve. And we do this very slowly in a very controlled way. And with that, it's also very easy for us to approximate where is the point of diminishing returns, where more data doesn't help us anymore. And we can also see where exactly the limits are, where we cannot get with the current methods that we are using any better results, even with more data. Does that answer your question?
Yeah. And how many data at the end you pull together, let's say, the ideal result? How many?
Right. So as you see on the setup, we had around, but typically we have between 50 to 100 plants on a trade finder, so in 2 square meters, and we scanned it about 10 times. And so that's pretty much the only training data set we need to generate with our setup to set up this whole pipeline.
Thank you.
Thank you, Andras. Our next speaker will also be talking about 3D phenotyping at the cutting edge with Xray CT. Our next speaker is Bas van Eerdt.
Hello, everyone. First, I would like to start with thanking the organizing committee and the scientific committee for allowing this talk today, and also thank you for hanging in there up to now. And the coming 20 odd minutes, I would like to introduce you in the magical world of Xray CT, which I'm quite enthusiastic about, to be honest. So my name is Bas van Eerdt. I'm founder and CEO of Phenoky. We're based in the Netherlands. And besides that, I'm also an Xray enthusiast. And there's one person to blame for that. He's over there. That's Mr. Stefan Gert from Fraunhofer Institute, whom I bumped into 10 years ago on a phenotyping conference. He was presenting a paper on Xray CT and potato tubers. And then I got hooked and got more and more excited about this technology in the past years. Today I'm also presenting on behalf of some other partners. So that's Fraunhofer IZRT from Germany. That's part of the Fraunhofer Gesellschaft, one of the largest research institutes from Europe. And they're dedicated to developing Xray CT hard and software solutions, in this case specifically for the plant and agricultural domain. And also Phenolytics, which is a spin-out from the breeding company Strube, who are the original inventors of the Phenotest concept, on which I will present a bit more later. And jointly we are focused and dedicated on developing and integrating Xray CT solutions, applications in the breeding and plant science domain. So let's start with Xray CT. What are we talking about? So it's very basic concepts. You have an Xray CT source that emits X-ray radiation. You have a detector that detects the radiation that passes through the object. We put an object in between, we rotate it, and the resulting beams that are not absorbed by the object are detected by the detector, and that's your signal, and that creates a 2D image. And while doing so, we create somewhere between 600 and 1600 images for every individual scan, which takes somewhere between 2 and, let's say, 15 minutes. It's quite a data beast, so you generate somewhere between 1 and 20 gigabytes per scan. And it's very high resolution. So your average resolution is around 100 micron, but we can also go down to 2 or go higher if possible. Then we have 1,000 images. They are in an automated pipeline inserted, automatically reconstructed in a 3D volume, normalized to remove some artifacts that are in there. And then the fun part starts in segmenting the 3D volume for the objects of interest that you're interested in. When these are segmented, then the actual measurement starts, and that is to then measure and write down the trait information and make it available for further research. Why Xray CT? Because it's obviously not the easiest way technology to implement. It might also not be the cheapest one to do. But I think there are a few pretty convincing reasons to consider it, and I hope to be able to share that with you today. First, we can reveal what's hidden, so you can make things visible that are not visible. Think of roots. What not everyone knows is that Xray CT is actually a biomass measurement. We are measuring biomass. So it's not a proxy, you are measuring biomass because you measure density, you measure volume, combine the two, you have biomass. And what are we all after? And not in all instances, but in many instances, biomass. And this measures the real biomass. True 3D, so not 2.5D, also not only the surface area, but it's true 3D, true and true. So outside, inside, fully reconstructed, high resolution as mentioned, non-invasive, non-destructive. And we tend to say, and I hope to prove it in some of the next applications, that it's also generating ground truth quality data, which opens a whole new venue of possibilities if you think about it. So today I would like to focus on a few applications because we promised to show applications throughout the life cycle of plants. First, seedlings, root systems, and also some slides on yield components. First up, seeds. So here you see an image of a container filled with seeds, a few hundred seeds, which is scanned and reconstructed. So it's a 3D volume. From that, individual seeds are being segmented, and from those individual seeds, individual traits are being derived that you see on the right-hand side of the image. So it's about seed code, embryo, size, shape, position, starch position and components, for instance, but also internal defects like insect damage could be detected and quantified. And on the seed lot property, so on the whole batch that you're measuring, you can quantify metrics like 1,000 seed weight and biomass. You can also tell something about heterogeneity and the purity of your seed lot. So how many of the seeds in your seed lot are the same type of seeds and how many foreign objects are there, for instance. I bet that not everyone knew that you can measure this amount of traits using Xray CT on seeds. A nice example here. Here you see 3 sunflower seeds. On the outside, they have exactly the same phenotype, essentially. But if you then peel it down and look on the inside, then you see 3 completely different seeds. On the left, you see a well-filled, healthy seed, which will probably germinate well. And the 2 images on the right, they have different types of problems, a necrosis or a broken radical, which might or might not result in healthy and fully developing plants. And so that shows definitely the benefit of looking inside the seeds and not only the outside. When you do the scanning and when you collect information, with that information you can also tell something about the quality of the seed. So you see well-developed seeds that will generate healthy plants, for instance, and you also damage seeds that might not result in healthy plants. But what you can also see is that, for instance, outside shape or volume of the seeds is not directly dictating whether or not the seed is healthy or not. On the top left, the 2 seeds are both healthy, but one is much smaller as the other, but both show a well-developed embryo, which is probably resulting in a healthy plant. Next up, germination testing. So probably you're all aware of germination testing that has to be conducted, sometimes in soil, sometimes in paper. And in many cases, the paper testing is the method that you have to use, and that's used in all companies. It's all manual, it's very labor intense, it's all done by humans, so it's very subjective and prone to errors. And the result of a trial of 2 weeks with 400 seeds, with people doing all the work, is often not more than 4 or 8 data points. Germinated, not germinated, normal, abnormal, and maybe you do the counting twice. So very little data points for a significant amount of work. So there's a new method available, and that all evolves or revolves literally around this spot. That has been developed by a breeding company, in a breeding company, together with Fraunhofer Institute. And that's what you will see in the next small minute. So this is a fully automated concept of automating the germination trials in vertical pleated filter paper. So this pot contains 50 seeds, and that will be automatically imaged using an Xray CT scanner that you see here in view. All the images are acquired, 3D reconstruction is made, all within the pot, without opening the pot. The paper is removed, the seedlings are isolated, and you have 3D ground truth models of every individual seedling that's in the pot. Those seedlings can be segmented, all the organs are identified, roots, cotyledon, hypocotyl. From every element you know the biomass, the length. When you have multiple time points, you also know the dynamics. So pretty powerful. On the right, the bottom right, you see all the traits that can be extracted. So what can you do with that information? So for instance here, this is just the basic information. So on the left you see normal developed seedlings. In the middle you see abnormal seedlings. And on the right you see non-germinated seeds, obviously. Also the abnormal detection can be done automatically by the software, and that can be configured based on the crops and also preferences of interest that the user has. And by the way, this technology is already in use for 5 years within this breeding company for routine work, for routine screening. So it's not in a lab, it's actually used day to day. Here you see time series. We follow a single seed over time, and you see the plants developing in 3 different seed lots. In the end, the germination count of these normal germination trials will result in the same germination observation. So they're all normals probably, and they're germinated. But you see that there is a completely different dynamic in how they germinate, and that can also be nicely now quantified on organ level, because the bars on the bottom represent the biomass per organ. Also, you can derive a lot of information about heterogeneity or the homogeneity of your plot, of your seed lot, which is pretty important if you want to sell the seeds to your customers. You want to know how they perform. And here you see 2 seed lots, exactly the same outcome from a traditional germination trial. But here you can clearly see the heterogeneity within the lot, and this can also be quantified in the output data points that you will receive. Vigor. I think the biggest on top of the list of all breeders is if you talk about seed quality and defining quality, is to learn how your seed might perform under stress conditions. And then also vigor is an important thing to quantify that. So here you see 3 different seed lots, again with the same germination performance, but with a completely different vigor behavior, which can be nicely quantified because you have all the underlying data points. Another example of what you could do with this is, as you might know, seeds are being treated with fungicides or other stuff to make sure that they perform well in the field. But the formulation of that coating around the seeds can be pretty precarious. You have to be careful in what you're doing because it can help your seeds, but it can also destroy your seedlings at the same time. And there's also multiple effects, so it can maybe boost germination, but maybe it can retard the development of the roots, as you see in this example. So this is an example of different treatments that above ground have similar effects, but below ground they result in a strongly reduced root length of the seedlings, which is important information to take into consideration if you are developing these formulations or if you want to quantify these. One more thing, I always wanted to do this. So we now talked about seeds, we talked about seedlings, but what if we combine the two? Then it gets really exciting. So here we have the pots, we can isolate the seeds. Before germination, we can already look into the seeds and quantify the organs in there, so we know what the seed properties are. Then the seeds are being germinated, and you see the development over time. That's the perfect base for modeling. So you know exactly on seed to seedling basis how a single seed develops into a single seedling. And when you have that modeling, you can also apply that on seed measurements. So you only measure the seeds, and you can more or less predict what the resulting seedling will be. And that can be pretty powerful if you talk about seed operations, where you have to make a lot of steps in the process from receiving the seeds to shipping them, where time is very critical. If you can exactly predict how it will perform, what your quality is, then you can very directly direct your processing steps and also calibrate your machinery to do so. So this is pretty cool, if you ask me. I'm a bit biased, I have to admit. But that was probably already a bit obvious. Another thing you can do with that is learn how what kind of seed traits result in what kind of seedling effect. So here you see on the left-hand side, you see high-quality seeds from all instances, and on the right you see seeds with issues and also the resulting seedlings. And and this is pretty valuable information to know because in seed processing, sometimes seed lots are being sorted by optical sorting or Xray or fluorescence or whatever. And then it's pretty helpful to know what are your good seeds and what are your bad seeds, and also to make decisions and to program the settings into your processing machines. So you can also use the data to make processing decisions to get the maximum potential out of your seed lots, and also to calibrate the performance of your machinery and to dictate the settings. So enough about seed and seedlings for today. Let's talk about roots. That's a very obvious application, of course, for Xray CT that has been presented upon already a few times here. But it remains a pretty powerful method to extract root traits from plants in pots. So you can do it from pots filled with soil. And of course, there are different soils that will yield different performances. But in general, there's quite a bit of things already possible with different soil types. You could extract information about the roots themselves, the architecture traits, biomass traits, and for instance also elements like root diameter. And again, it's non-invasive, and it's also enabling multi-time point observations. So when you have multi-time point observations, you can obviously track the development of the root system. So here you see an example of a maze experiment where the black images on the bottom are taken on a 7-day interval, where you can clearly see, and again, it's behind it is 3D data, and we are looking at a 2D representation, but you can clearly see the increase in biomass over time from the root systems, which can be valuable information. On the right, you see a different data representation with the different time points that have been measured, and there you also see the distribution of root volume over the depth within the pot, which might also yield interesting information. Another example is this experiment with sorghum. So it was a control plant on the left versus a stress plant with limited nitrogen and water. So you can already in the washed root systems, you can see a clear difference in the root biomass, and you also see that represented in the Xray data that you see in the black rotating movies. But you can also see that the architecture is different, so you can learn a whole lot of information that will otherwise be very difficult to extract when you have to depend on root washing or on other 2D approaches. Yield components. So that's in the end, everyone wants to learn a lot about yield, and also about yield is affected during the development of plants. So we're approaching the end of the life cycle of plants here. So but this is a nice example, and that was the the poster that got me excited, but it's still a nice piece to be able to present. It's on the imaging of potatoes using 3D CT imaging. And here the images are taken in a time course, and it's used to determine the biomass and individual potatoes in the pots. Measurement time approximately 4.5 minutes. So what can you learn from that? Here you can see on the bottom images the development of the tubers, which is already pretty nice to see how that develops. But it's not only the visuals, it's also the data. So all tubers are segmented individually, and also the biomass of all tubers are quantified automatically in that process. So what you can see on the top graph on the right is that it's correlation numbers between dry and fresh weight. So the fresh weight is what's actually being measured by the Xray, but with a certain calibration, you can also very accurately, as you can see in this instance, derive the dry weight from that. And on the bottom, you see the tuber weight development over time of the individual tubers within the pots of that have been X-rayed. Next up, wheat ears. I learned yesterday that wheat is the dominant crop that everyone's interested in the phenotyping domain in some kind of poll that was specified. So what's being done, and this is actually an image of the system that's in Adelaide at the Plan Accelerator. And that's being used for doing high throughput measurement of wheat ears in a non-destructive manner. So different wheat ears from trials are being placed in vertical tubes on so-called carriers, around 30 apiece. And those are being scanned automatically in a batch by the PhenoCT, as we call it. In a few minutes' time, the entire object is scanned, and then the individual wheat ears are segmented from it. And then from those wheat ears, we can isolate the individual seeds and seed traits. So here you see what's happening. On the left, you see an isolated wheat ear. Then on the image next to it, you see in colors the different identified seeds in the wheat ear. And if in the middle, you see a representation of all the seeds that have been identified on the wheat ear. And on the right, you see all the parameters that are being extracted from that, which is on spike level or sometimes on single seed level. Again, biomass, shape, and position. And what you, for instance, also can do with that is that you can quantify whether or not seeds have been aborted, or that they have been underfilled, or that they are properly filled. And that can be interesting information to not only specify the number of grains, but also the biomass and the total yield of the crop. And on the left, again, you see that there are pretty strong correlations with drought stress and drought and heat stressed examples. So it's a pretty robust technology that you can use in high quantities. And then the last example, a bit of an odd one. If you talk about yield components, then for trees, wood is the yield. So and the people from Fraunhofer are conducting also field trials with doing CT scanning of mature trees, where you can quantify of course the biomass, but also the type of wood within the tree. So whether it's harder wood or softer wood, that can be nicely quantified using this method. So I hope that the past minutes I could have put these words to the test, and that demonstrated that all that's stated here, that it holds some merits. And if you are getting enthusiasts, which I can of course imagine, then there are different ways to proceed. Then you talk about hardware. There are different elements. You can think about tabletop solutions, intermediate solutions, and large-scale solutions, as you see on the right, which is an example from Purdue University. But together with the partners that I mentioned, we can not only build your equipment, we can also do proof of concept studies, validation pilots, and can even, in collaboration with Fraunhofer, you can develop completely new methods and equipment if you want to. And I think there's a lot to explore yet in this domain. And maybe one last message is that Fraunhofer Institute is also establishing a new test center that's being constructed by Phenotyping at the moment, which will be ready by the end of Q1 in 2025. And that will be a perfect testing ground for testing out all sorts of different applications for Xray, but also for other sensor technologies. But Stefan Geert can tell you much more about it. So that's all I wanted to share with you today. I hope I've been able to share a bit of my enthusiasm, and I'm very happy to answer any questions if there might be. Thank you very much.
Ambassador Richard Dick from the APIC. Just in the field systems, the human safety aspect, I mean, how is that managed?
Human safety, you said?
Yeah, you've got X-rays flying around.
Yeah. Good question. That's the important question. So all devices are intrinsically safe, so they're fully X-ray shielded and protected with interlock systems that mean that the sources will shut down immediately if a door is opened without permission. So the remaining radiation outside of the unit is equal as the ambient radiation.
Bas, my question was about the field systems, like when you're doing trees.
Sorry. Sorry. Apologies.
The operator and so on.
Yeah. So in this case, it is a remote-operated vehicle. So this is a very early prototyping stage. So this is more proof of concept on the method, and then it's just by using a certified remote that you can be at a safe distance, that you're not exposed to X-ray radiation. And in the future, that might be and will be implemented in a more intrinsically safe manner, that you can actually drive something and be close to it.
So it's amazing what you can reveal, what is normally hidden. My understanding was that if you do a CT scan one time on an organism, that's quite fine. But if you do it a number of times, you see retarded growth. Yet you show that you follow your plants over time. Have you tested what happens to a plant if she gets the amount of doses that you apply?
Yeah, thank you for the question, because that's a question that many people have. Two answers. So you see the example of the breeder Shree. So they are using it for routine screening for the germination testing. And before they made the final call in switching to this kind of testing, they did very rigorous testing to make absolutely sure that there is no effect caused by the X-ray radiation. So that was validated on that way. And in the potato experiment that was also demonstrated, there was also it was a duplicate test. So one set of the plants was regularly exposed to X-ray and the other one not, and there were no visible effects.
Right. So does that mean that there are some species that are more sensitive than others?
I cannot. We have not observed ourselves any kind of influence on the development of plants as a result of X-ray radiation. That's the answer I can provide you. I cannot provide a empirical answer on that nothing is affected, because we have not tested everything, but everything that we have tested and have been using in X-ray has not shown any kind of effects caused by X-ray radiation.
Good.
Regarding the germination test seeds, have you ever tried or tested on seeds from a species that has polyandrony, polygenic species?
Sorry, that has what?
Have you ever tested this equipment on seeds from species that presents polyandrony, more than one embryo within the seed?
Yes, absolutely. So in sugar beets, that's a common. That's where the technology was developed. So multi-germ seeds, if that's what you're alluding to, that's a common thing there, and that can be quantified in these instances. Yes.
We have time for one last question over here.
Thank you. Quick question. Root imaging, how important is the substrate and the water content in the pot?
Yep. Also, again, good question. Thank you for that. So as mentioned, we can. It's not that you don't have to use artificial soil. So normal soils can be used, but there is that depends, of course, on what the definition is of normal soils. So we have had quite tricky soils where we were able to extract root systems pretty well. But we also, if you have really, really tough soils, then it can be a bit more complex. And additionally, the water content that you mentioned is something that we also see that can affect the segmentation, but it's all about the combination between crop, soil, and water content. But it's definitely important to look at the protocol to make sure that you can get the best results out of it. I think.
Thank you, boss. Now we have our flash session. So if all the speakers in the flash session can kind of come up here and get established here. And this is going to run very quickly. First up, we have Kirby to Jode.
Good afternoon, everyone. I am Kirby from Heuristic. We originally started as a hardware company for smart glasses, but along the way, we decided to shift more to a full solution company. Because from one of our colleagues, Maarten van der Stukken, who is now back to his first big love, research, and our partnership with Bayer, we learned that there was a real need for phenotyping applications. So after diving a bit deeper into it, we started to develop our own data collection app. We developed a web portal where you can configure your observations and an app that works on your cell phones, tablets, and with our smart glasses to run your crop screening sessions. So once you've finished collecting your data, you can go into the app or the web portal and view it, or you can export it. Combining our app with our smart glasses allows you to operate in a hands-free manner, making crop screening a lot less time-consuming. Working like this, by freeing up both hands, so that you can, for example, pull open a crop and check if there are any diseases, and using voice-based data entry results in up to 55% efficiency gain in crop screening data collection. Having the data instantly digital results in up to 20% less human errors, and having the ability to have flexible but structured workflows results in up to 25% increased data consistency. So if you want to learn more about our app and our smart glasses, I invite you all to come talk to us at the upcoming poster session or at our booth that we share with Vito in the exhibition hall. Thank you for your attention.
So hello, I'm Pierre Lejeune from the University of Liège in Belgium. So I'm really glad to be able to present this project, APIC. In French, APIC means something like to be at the right time at the right place. So you might say it's just yet another phenotyping platform. This one has a few specificities. It's meant to be a qualitative phenotyping platform for optimizing high added value production. So how does it do that? So basically, on the upper side, you can see it's a mobile gutter system, 2 stages on top of each other. At one end, there is a hyperspectral imaging station with which we can develop some predictive models. In this example, this is polyphenols in lemon balm. At the bottom left of the slide. On the other side of the system, there's an irrigation station that can perform gutter-wise fertigation, which means that every gutter can receive a different nutrient solution potentially. And the system is a conveyor that cycles the plant at least once a day. And it has also luminaries, LED luminaries with 16 channels that can be pre-programmed to deliver any recipe that you want. And although the luminaries are fixed, the light can sort of follow the gutters as they move in the system. And so we can do also some light optimization experiments with light gradients. And the final goal is to optimize biomass, for example, in medicinal plants, in terms of yield and quality. So if you have further questions, the poster is number 42, which means it might have the answer to everything. Thank you.
So, hi everyone, I'm Fabien Miart. So in the last decade, there has been a lot of development in the field of high-throughput plant phenotyping, especially with robotics and AI. And most of this development have been applied in the field or in indoor platforms. But why this development have been developed in field and indoor platforms? There is still one phenotypic scale which is largely missing from the game. I'm talking about the microscopic scale. So the main reason for that is that the HTMPP and cellular biologists are not talking enough together. So to help bridge this gap between HTMPP and cellular biology, I have launched MIAtecs this year. So let me introduce you with HTMPP for high-throughput microscopy plant phenotyping. And the objectives of MIAtecs is to bring you with new solutions to help you find hidden phenotypic traits from the seedling scale to the subcellular resolution. So the MIAtecs services pipeline is in 4 steps. First, we help you find the best imaging solution and develop your own device. Then we help you find the best microscope that will fit your needs. We also analyze your images or develop image analysis solution that perfectly fit your needs. And finally, we develop advanced analytics tools with graphical unit interface, graph statistics, and visualization. So what are the benefits of HTMPP? You can, with our solution, you can image the shoot and the root part of the seedlings at the same time and compare what is comparable between multiple seedlings. Then you can access the gene expression at the microscopic scale along the seedlings. So you can image, for example, biosensors like pH, calcium, ROS, or phytohormones. You can apply chemical treatments very precisely and at low cost, so in an eco-friendly way for your pesticides and chemical products. And you can also, from the early phenotypes, maybe predict what's happened at the plant scale level, so to help you analyze the yield and stress responses. So come to my poster number 41 to discuss a bit in more detail what MIAtecs can do for you and to introduce you with this small microscope here.
So good evening, everyone. So I just want to give a little bit of brief of the work that we do in a Salk Institute. So the Salk Institute program called Harnessing Plant Initiative, which actually try to develop a plant which can have the larger and deeper root system that can store the more carbon in the soil. So we actually wanted to use the plant power to address the climate change. So how this works? Plant takes the carbon dioxide, sends to the carbon forms, and via the root, it stores into the down. So to do that, we have developed quite some multi-modal phenotypic platforms. Sorry, it doesn't go. Oh, yeah. So I'm not going to go detail about all the methods. Still not working. Sorry for that. Okay. So these are some of the platforms that we use. These are automated best pipelines, and I'm not going to do on that. What it does is actually it gives more root-related traits up to 1,000 different traits, and we automated all those pipelines to get those data. Using those data, we use the genome-wide SSS mapping and identify the genes, and we also have in a Salk in-house, we created either mutant or overexpressed lines, and those lines are used for the validating those traits using the larger phenotyping modalities. And yeah, so we use those modalities just to validate those lines in a larger pod at the flowering stage before that can actually lead to the translation to the field. So you can see one of my colleagues with the root head. So if you want to know more about our work in progress and what the future holds to use the plant to address the climate change, please visit my poster number 43. We will have a fun talk with a coffee. Thank you.